I hope it is visible.
It is visible. You can start.
Right. So, good afternoon to one and all. Thanks to the organizers for providing me the opportunity to be part of this conference. And the title of my work is Antioxidant and Antimicrobial Activities of Phenolic Compounds from Tecomella, Undulata. So, since time immemorial, human beings have always aspired for good health, happiness, and immortality, knowing that it cannot be a true story every time. However, plants have played a pivotal role in achieving these goals to a greater extent. In fact, the therapeutic potential of medicinal plants have been laid down in ancient systems of medicines, including Ayurveda, Siddha, Unani, Chinese, and so forth. And mainly the bioactivities of plants is basically due to the presence of secondary metabolites, which are not needed for the growth and development of the plants, but have ecological role, ensuring the survival of the plant in its ecosystem. No, such is promising, lucrative, and important is this field of science that one of the data suggests that till 2050, the trade of medicinal plants will increase to the level of 5 trillion USD. So, in my opinion, that is nothing secondary about secondary metabolites. However, like we have two sides of the coin, every aspect has pros and cons. So, since these plants are very much commercially important, they are overexploited, and that is one of the reasons that every year many plants are being included in the red data book. So we need to have an alternatives, and plant tissue culture can be one of the effective ventures in this regard. Coming to my experimental plant, Tecomella undulata from the family Bignonaceae, and it is, as you can see in figure B, it is native to India, Pakistan, Oman, and adjoining countries. And it is also part of many Ayurvedic and herbal formulations. Now, ethnomedicinally, it is used to treat hepatic disorders, skin disorders, and mainly disorders which are caused by microbes. In fact, one of our research publication, which has been accepted in Frontiers in Pharmacology, we explored that in India, mostly the stem bark is used. And in Pakistan, stem bark and flower is almost equally used to treat disorders. This is a plan of work. First of all, we collected the plant and took some explant to generate callus culture. And then both the samples were sequentially extracted. We started with methanolic extract and sieved it to ethyl acetate fraction, which was further used for bioactivities and metabolite profiling. Now, this is the table which shows comprehensively the tissue culture part. In fact, I have removed a few of the combinations because of space crunches. You can see that Morashid and Scooge media was used, and individual combinations of phytohormones did not induce much of callus, but the combination, that is NA and BAP and IA, induced significant amount of callus. Similar pattern was observed when the callus was induced through nodal explant, and then we finalized that the leaf generated callus will be taken further for research purpose. Now, this slide shows the development of callus from leaf, and the figure D shows basically the subculturing part. This is with respect to individual hormones. Now, here the hormones that clicked, shown in green, BAP-NA and BAP-IA. You can see that a good amount of callus is there, and it is friable so that it can be used further for suspension culture. Now, these are the slides showing generation of callus using nodal explant. In fact, BAP showed callusing and simultaneously organogenesis. But since we were focused on callus, we took again the combination showed in green. Now, when we had both the samples, the plant parts and the callus culture, we went ahead to assess the bioactivities and phytochemicals. The first one, that is total phenolic content, and the other was total flavonoid content. In fact, the maximum content of TPC was found again in BAP-IA, that is callus combination. Similarly, in TFC, we saw that callus took the lead. Only comparing the plant parts, the maximum content was seen in stem bark, TPC, and in TFC, it was flower. Now this slide shows various antioxidant assays that we tried to ascertain whether it has some antioxidant potential or not. We took DPPH, nitric oxide, ferric reduction, and deoxyribose degradation assays. So talking about the DPPH, it's not moving. I don't know. I'll just see. So talking about DPPH, it showed that IC50 value was pretty less. In fact, and lesser the IC50 value, better is the antioxidant activity. Again, talking about nitric oxide scavenging assay, similar pattern was used. And in ferric reduction, here we see how much the potential is of the extract and the phytochemicals to reduce ferric to ferrous. So more the ferrous production, better is the activity. So we found that here again callus was taking the lead. And talking about the IC50 value of DDA assay, again callus. So overall we found that callus had the better potential. Right? Is it visible? I think.
No, it is not visible.
Is the slide visible?
No, it is not.
Okay, I'll again share them again. Let me just check it out.
Dr. Shad, should we run the slides from our side?
You can.
Okay, all right. Jean, can
There
you?
is some technical glitch, I guess.
Please mention the slide number.
Just a second. The slide number is 13.
Okay.
Right, this one. Thanks a lot, ma'am. So here we assess the DNA damage inhibition, and we took PBr322 as the DNA incubated with hydrogen peroxide and irradiated with UV. So that you can see in lane C, the whole of the DNA got degraded. And once we put the DNA with the extract and irradiated with ultraviolet rays, we found that the flower and the callus both actually helped out in inhibiting the DNA damage. Next slide. Next slide, please. Yeah. So since we got initial good results, we thought of analyzing the phytochemicals present in the extract. So this is the chromatogram that shows the standard compounds which have been assessed. In fact, most of the compounds have been reported for the first time in callus and plant parts of this particular tree. Next slide, please. Yeah, this chromatogram shows the profiling of different plant parts and callus culture. Next. Now this shows, as you can see, the individual contents. So in flower, maximum content was that of naringin, and all the green representation shows the maximum content, and the red one showing the minimum content. Comparing the callus with that of the plant parts, we found that callus had more content of individual phytochemicals. So that was again quite good, so as to have an alternative approach to harvest metabolites from this plant. Next slide. Now, since ethnomedicinally we found that antimicrobial activity is quite good of this plant, so we use Pseudomonas aeruginosa, which causes respiratory infection, skin infections, UTIs, and other infections as well, because these organisms are able to form biofilm, and that leads to higher resistance to antimicrobials and higher pathogenicity. Next slide, please. Next, yeah. So we first of all did the preliminary analysis using antibiotic susceptibility assay. So left side you can see that there is inhibition zones, and on the right side you can see that again here stem bark had the maximum activity against these organisms. However, other samples were almost at par in terms of the activity. Next, please. Yeah, so we went ahead analyzing the MIC, and it is suggested that minimum inhibitory concentration of any sample, if it is lesser than 100 mg per ml, can be a good candidate to assess the antimicrobial activity. So you can see here, most of the samples had good activity, and the leaf and again callus generated from leaf had the best activity. Right side shows the quorum sensing. In fact, what happens, pseudomonas grows, and once a particular number is reached, they again behave like a quorum. In fact, quorum gets achieved and they behave like a group, and that leads to lesser activity against kind of any antimicrobials. It increases the pathogenicity and virulence as well. Next slide, please. Now this shows the activity of again the samples. Basically, you can see other different. We took actually sub-MIC concentration. Sub-MIC means MIC level was taken, and then we took all the concentration lesser than that. And you can see, as compared to the control, the absorbance got quite decreased in all the samples that we analyzed. Next slide, please. Now this shows the figure of pseudomonas. In fact, we took a glass slide having well so that we can inoculate pseudomonas so that they can form a film. In fact, the figure A shows several pseudomonas cells. We actually saw the cells by intubating it with crystal violet. And once we put the extract and then tried to see the biofilm formation and number of cells, it significantly decreased. Again, the leaf extract and BAP-IA combination took the lead. Next slide, please. Right. So here, what happened once we got again promising results, initially we thought to further analyze and to confirm the antimicrobial activity. So we took pyocyanin. So pyocyanin is a kind of pigment, blue-green pigment that is produced by pseudomonas. So lesser the content of pyocyanin, more is the activity of the sample as an antimicrobial. So you can see in all the figures comparing to the control, wherein we didn't intubate pseudomonas with the sample, the absorbance was close to 1. And once we treated it with the samples, it went and reduced considerably. In fact, 5 times almost the pyocyanin production decreased. So this again suggests that somehow our sample and the phytochemicals present in the sample is playing adversely with respect to pseudomonas and its metabolism and inhibiting its growth. Next slide, please. Now, since we had a kind of good results, and since the plant is endangered as well, it is there in the IUCN Red Data Book. So we need to have some alternatives to again save this plant as well. So we used elicitation in the suspension culture to have enhanced production and harvest of the metabolites. Next slide, please. So left side shows the sigmoid growth curve of the suspension culture, and the right shows different stages. That is again lag, log, and exponential. So we established a suspension culture and harvested the metabolite. In fact, analyzed the metabolite at different stages. Next slide, please. Now this table shows that we used methyl jasmonate as an elicitor at different concentration, but the best concentration clicked was 100 μm, and the time of harvest was 72 hours. This suggests that elicitors can be used, but we need to be very sure about the optimum concentration and also the time of harvest so that we can have more and more content of metabolites. In fact, it is important to mention over here that certain compounds like naringin, syringic acid, which were not present in the control, also showed in 100 μm concentration, so that we can somehow assume that it triggered the biosynthetic pathway for the production of metabolites. Next slide, please. So concludingly, as I mentioned, it's very medicinally important tree, and there are several first-time reports in the present study, like comprehensive callus culture, bioactive phenolics from the plant parts in callus culture, the Pseudomonas aeruginosa antimicrobial activity, and elicitation part. In fact, overall, we concluded that callus had more number, content, and bioactivities as compared to the plant parts, which was promising. But still, we feel that further studies are needed, taking the lead from ethnomedicinal importance of the plant and validating it scientifically. And we need to be sure, and we need to think about alternative approaches also to harvest the raw material and the metabolites from this plant. Thank you, everybody. Next slide. So any queries, if any, are welcome.
Yeah, thank you, Dr. Sharat. If the participants have questions, they could share it in the chat box.
Right. Thanks a lot.
It is a nice presentation. Please, before